Good evening, everyone. This is another opportunity to meet at Omega Bio-Edutainment, where we will be so much focused on what we have done so far and how can we actually prepare for examination. Now, I want to center our point of discussion this evening on microbial genetics. It's going to be centered on microbial genetics. Okay. And I want you to focus on the following topic, so to say. I want you to focus. So far, so good. What we have so far has just been that altogether we have more than 10 videos that is actually dedicated for this this. And this video, we are going to focus the exam is going to focus on the following. uh, don't forget, the first thing we did was to look at gene expression and gene regulation. So, and don't forget that if you want to really pass the exam very well, you need not to focus only on what you have learned so far on Omega Bio-Edutainment. So anything that has to do with gene expression and gene regulation, I want you to focus on it. Then we are going to also focus on transcription as a process. Transcription as a process, especially in bacteria. Transcription as a process in bacteria. Then after that, I want you to know something about the mutation. We know I devoted a lot of videos on mutation. So we will have to look at the molecular basis, and after that, you should be able to give us some examples and case studies. Examples and case studies. Then again, don't forget that at this level, um, each question will not carry more than I think either between 20 and 25 marks. And if you are uh, if you are so all I'm doing, I'm very sure that gene expression, once you are able to do anything very smart on that, you know how much mark is going to wait for you. Then after that, I also want you to center your point of discussion on conjugation. So you need to focus also on conjugation. And what we mean by conjugation is on genetic. So we need to focus on gene transfer, how gene are being transferred. So gene transfer. So, and gene can be transferred through conjugation. That is the one that has to do with sexual means of gene exchange. So don't forget, there are two basic types of how gene are being transferred. We have horizontal gene transfer, and gene can also be transferred through vertical. If we say horizontal is when you have two species, the same species. If this is organism A1, organism A2, don't forget that two of them are A. So if there is a gene of interest from here and it is transferred from this one to this one, that is called horizontal gene transfer. But when we now have a gene of interest, if you have a gene of interest in the parents, in their parents, and that gene is now going to be transferred to the um, offspring, is going to be transferred to the offspring, that is called vertical gene transfer. So the first method is through conjugation. The second that you also have to focus on is transformation, transformational principles. How genes are inherited or how genes can be transferred by the use of a technique that is called a transformation. The other one is by transduction. That's another one. Transduction, the one that's to do with phage, phage transfer. All of you know what a phage is. That is from bacteriophages. That is talking about bacteria viruses that feed on bacteria like E. coli. So if there is going to be any gene of interest or exchange of genetic material between the virus and the bacteria, it's going to be through the process of transduction. Then after that, we have mentioned. So you need to also know something very, very importantly about gene code or genetic codon. Genetic codons. You need to know that. Don't forget how I dedicated certain videos for you to be able to learn the genetic code by heart so that you don't need to actually cram them. What you need is to know the simplest principle by which you can remember all the 64 amino genetic code. So I said 64. Out of these 64, 3 are called stop codon. Then one is known as a start codon. That is a methionine, and the genetic code for that is AUG. Now, having known something about that, I also want you to also focus on my video. On my video that is so one that is dedicated for this, one that is dedicated for this, one that is dedicated for this one, mutation, this one and this one. So by now I'm very sure that you have nothing less than between 7 and 10 questions, between 7 and 10 questions, out of which, if you are so clever, you will be able to know um, what and what you should actually focus on. Now, I want you to also know it is worthy of it is worthy of you to note that how can you, if you are given any questions like this, what are those things that is expected as a microbiologist or as microbial genetics? So what are those things that are that are necessary, and what are those things that are not necessary? Don't forget that you should not just go straight to point. You don't need to write stories. Don't need to write story. You should be able to give very, very precise, succinct explanation. For example, let's say asked to you are asked to write a note on how gene are expressed, that is gene expression, okay, and how is this gene being regulated. We have to be picking it one after the other. Gene expression and gene regulation. Don't forget, what you should do is, the first thing is, what is gene expression? What do you understand? How can you define gene that is being expressed, and what are those genes that must be expressed. So you need to talk about the definition. You define it for us. You need to talk about the importance of gene expression. Then you need to tell us about the detailed mechanism, not too detailed like that, but you should be able to know detailed mechanism. And what do we mean by mechanism? Mechanism is such that if this is a DNA, don't forget the whole thing of gene expression is from the journey of DNA into RNA, then later into protein, which is going to be of interest. So whenever there is a complete journey like this, that is the journey at which protein is being expressed, or any gene that is being expressed. So here we have the process is called transcription. So all the mechanism of DNA transcription, I am expecting that at this level you should focus on the videos and topics that can help you to be able to have a proper understanding of the process of gene or genetic transcription. So, and this one has to do with RNA polymerase as an enzyme. RNA polymerase will play a very vital role in that. Apart from that fact, from this is going to be transformed into messenger RNA. So messenger RNA, transfer RNA, and ribosomal RNA. You need to know that. Now, if you now know messenger RNA, don't forget the messenger RNA is like this. Okay. The transfer RNA is like this. Okay. On top here, you have amino acid ends. Here, so that is here you have anticodon. Okay, so if this amino acid is coding for methionine, so what is going to be here is going to be AUG. Don't forget that the fact that you now have is telling you that it has been converted from DNA into RNA. That is why you have U here. And don't forget, I told you very, 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 very importantly, you should learn by heart. It's not a matter of maybe you cramming it, but at least you should be able to learn by heart all the 64 genetic codons, so that whenever you need them, the first point of call should be your where you must have written it to. Go back to the same video that I asked you to decoding the genetic code. There is a special video that is dedicated for that. Now, transfer RNA, and out of all this, the cooking pot, that is where this one is taking place, is called ribosomal RNA. Nobody can cook beans without a pot. Don't forget that RNA or ribosome is the cooking pot, and this is a process that is called protein synthesis. So, and that is called the principle of translation. Okay, so this is a double-stranded. This one has become single-stranded, and this one will now be in form of a amino acid sequence. Now, the whole process is what is called gene expression. And now don't forget that there is difference between how gene is being expressed in prokaryotic organism and how it is being expressed in eukaryotic organism. If we are talking of eukaryotic organism, that has to do with organisms or cell that have organized organelles. That is, they have membrane-bounded organelles at which all this can actually take place. But in the case of bacteria, which is a prokaryotic organism, the level of gene expression in that is much more simpler than what you have in eukaryotic organism. Then don't forget that in eukaryotic organism, we do not actually talk of we do not talk of what is called post-transcriptional modification or post-translational modification. So what we have solely is immediately the gene is being expressed, that gene will just be processed and it is going to be start functioning. So the production of a functional gene, if they ask you to define gene expression for you, it is a production of a functional gene from double-stranded DNA through the process called transcription in the presence of an enzyme that is called RNA polymerase. The first point of call is going to be the formation of a ribosomal, it's going to be formation of a single-stranded RNA, which could be messenger RNA, transfer RNA, and ribosomal RNA. So, to the process of translating the information or the genetic code that is already encoded in the DNA into a gene or a functional protein. So that is the whole process of gene expression. That is how gene is being expressed. So another thing is, how can we know what are the importance of gene expression? The importance of gene expression to a certain uh, microorganism, be it prokaryotic or eukaryotic organism, is that any gene that is going to be expressed, that means the system will definitely need that gene. For example, and that's the reason why we now have non-essential gene and essential gene that are, so the genes that are variously that are expressed at almost every time means that that gene is very very important, very very important to that microorganism. So gene will not just be expressed for the sake of being expressing it. It is going to be expressed only when it is needed, and that is when gene regulation will now come in. What do we mean by gene regulation? Gene regulation is just a process of switching off, that is, of regulating the amounts, the timing of gene and the gene that is being expressed. So that means the gene must be regulated. I told you it's just like when you have a factory, very big factory, that what that factory knows to how to do best is just to be producing. It will just be producing. There's no time that production will stop. So you just discover that at every point in time, it will get to a time that the machine or the equipment and machine that they are using to be able to produce all these products will get to a point that it is going to wear out. It is going to knock because the machine must have overworked itself to the level that it will get to a level that it can no longer work. So the cell will never get into that point. They will never get into that point where gene expression cannot be controlled. So it is just like when you have a very fine car and that car does not have a brake. There is no time that the gene will be one way or the other is going to slow down. There's no time, nothing in that vehicle that will reduce the speed. So that is tantamount to the vehicle having an accident. So, and that is exactly what gene regulation and gene expression is all about. So please, you need to know. Go back to some of these videos where we discuss about how gene is being transferred. Then the next question is, how does the environmental factor, or how does what role do environment play in gene regulation? So for example now, so that is telling us is that the type of gene that will be produced or that will be expressed depend on the favorability of the environment. If the, for example now, if we have a situation whereby a cell supposed to produce glucose, if the cell supposed to produce glucose for respiration, cellular respiration, and already the environment does not favor the production of glucose. The environment favor production of fructose. So what is going to happen is that the environment will now force the bacteria, or is going to force the microorganism to be able to produce fructose instead of glucose. So that is what is called, that is how, or the role that the environment will definitely play in gene regulation and gene expression. So please, I want you to take note of this very, very well. Now, at this point, I've certainly talked about if you have any question like this, the first thing is that you need to understand the concepts. What is the basic concept of gene expression? You have to understand it from the primary point of view of the structure of DNA. How does the DNA actually look like? What's the difference between DNA and RNA? Then you should be able to talk about different types of RNA, and you should be able to pinpoint important points under transcription. So I just, like I told you, you don't have to start telling me stories. Make sure you bullet your points. Your point is being bulleted, just like this. Just very, very short and precise answer to all these questions. So that is that about transcription. That is that about gene regulation, or what is called gene what expression. Okay, so I want you to take note of that. Now, the next thing we will also do, that I also want you to focus very well on, is going to be on transcription. That process of transcription, I want you to focus on it very, very well. Transcription. And this transcription, you have to focus on it as transcription in bacteria. How do transcription in bacteria, how do it actually occur? Transcription in bacteria. Okay, so I'm going to basically, I'm going to tell you some vital points about it. Okay, about it now. Okay, I'm going to tell you. I'm very sure that by now nobody should tell you what to do and what not to do at this level, knowing fully that I've actually warned you, and I've just listed some of what you should actually talk about, some of those things that you should talk about and you should focus on. Okay, I want you to focus. So That is that about gene expression and gene regulation. Gene expression and gene regulation. Okay. The next thing, just the same way I told you, the same thing or the next thing should be your focus in transcription in bacteria. Transcription in bacteria. So what do we mean by transcription? Transcription is nothing but is the process by which the messenger RNA is a process by which messenger RNA, you know that the messenger RNA in the messenger RNA is being synthesized from DNA in the presence of an enzyme that is called RNA polymerase. That is the whole essence of what is called transcription. It is just a process by which the double-stranded DNA produces a single-stranded messenger RNA in the presence of an enzyme that is called what? That is called RNA polymerase. Don't forget to mention some of the important parts of transcription. You need to know what is called promoter region. Read more. Read more about enhancers. What are the functions of enhancers? You need to read more about what is called Tata box. Okay, focus. Go back to some of the videos that is dedicated to this on Omega Bio-Edutainment. So promoter regions, enhancer regions, and Tata box. So you need to do this. Then, having known that and having said that, at this point, the RNA polymerase will do two important, will play two important roles, very key. The first one is that they are the one that will initiate the process of transcription by binding at the promoter region. By binding at the promoter region in order for initiation to take place. Then don't forget this our transcriptional factor 1 to 2. Transcriptional factor 1 to 2. You need to know what their function is all about. So after they have binded on the promoter region of the DNA, okay, the next thing is that the process will commence, transcription will commence. After transcription must have commenced, now the next thing that is going to happen is that the RNA polymerase is going to unwind. It's going to unwind the double-stranded or the double strand of DNA. By the time that has unwinded it, the next thing is for messenger RNA to be produced. So on the unwinded section or on the unwinded strand, so what is going to happen is that the RNA polymerase is going to start replacing wherever there is a T, is going to replace it with U now. And by the time that has now been replaced, that is to tell you, that is the indication and implication that messenger RNA has been formed. So that is, it is no longer DNA. At this point, we now have a transformed process where the DNA has transformed into messenger RNA. Okay, so you need to know that. Then you also need to focus if you are talking of transcription process in bacteria. You need to also focus on comparison. How can you compare transcription in eukaryotes and transcription in prokaryotes. You need to compare. You need to know very, very well. And what are those things that you need to compare? You will just discover that inside the eukaryotic organism, because the eukaryotic organism has, in terms of their structure, they have a structure where you have organelles, specific organelles inside the nucleus that is taking place. So the what's going to happen is that the process will start of transcription will start from the cytoplasm uh, in eukaryotic organism, and immediately after the cytoplasmic initiation of this is going to also lead into translation. Okay, then I've said that cytoplasmic process, transcription is cytoplasmic. The location of transcription in eukaryotes is inside the cytoplasm. That is one. Then the second difference is that there is nothing like polyadenylation. Oh, sorry, there is a adenylation in eukaryotic, but there is nothing like a polyadenylation in prokaryotic organism. So that is that is, there is nothing like polymerization, capping and adding of A. Nothing. So there is nothing like a post. I have said it. Nothing like post transcription process is lacking, and post translation process is also lacking in prokaryotic organism, whereas in eukaryotic organism this is highly and very, very pronounced. So now I think the first thing, we have settled the issue of gene expression and gene regulation, and now we have also settled the issue uh, that has to do with transcription in bacteria. So, and how can we compare transcription in bacteria with that of a transcription in eukaryotic organism? Okay, don't forget. So, don't forget the process of transcription, that is from double-stranded RNA and DNA into single-stranded messenger RNA. That is process of transcription. And don't forget the function of the following: promoters, enhancers, Tata box, transcriptional factors. You should not forget their function. And don't forget the role that is being played by RNA polymerase in the whole process. That is very, very important. Now, the next focus, which I also want you to focus, is that focus on is, you will see that I dedicated a lot of videos on understanding the basis the molecular, the biochemistry of what is called what of mutation. Mutation. So you need to focus on what definition of mutation. What is mutation? Okay. After that, types of mutation. So you need to look at the molecular basis now. Molecular basis. How can you explain mutation in terms of from molecular perspective, molecular basis? Then you need to also know, which is very, very important, you need to know case studies, stroke examples, case studies and examples in each case. So you should be able to come up with an hypothetical examples or case studies that is going to be molecular in nature. So that one, you can use it to explain all these types of mutation, like mean sense, nonsense, frame shift, deletion, insertion, substitution, and so on. But don't forget that mutation is then what are the objectives? of us studying mutation at this point in time. What are the essence of mutation, or what are the roles played by mutation in genetic disorder? So, okay, roles of mutation in genetic disorders. Okay, genetic disorders and cancer. So, I told you that mutation primary is the primary stage, or the primary level of cancer is mutation. Okay, so we need to know that. Then, just like I told you, just like I told you, the issue of genetic code, genetic codons, you need to understand it very, very well. Please, very, very well. It is very, very important that you understand it. I gave you, I dedicated some videos on it. Okay, UCAG, then UCAG. Okay, so then this one you now write UCAG. I'm just trying to give you the hints. Don't forget that this lecture, this is not a normal lecture per se, but hints. So UCAG and UCAG. Please, you have to know very, very well. Know them very, very well. Don't forget, this is a 3 prime, 5 prime. Okay, 3 prime end like this, 5 prime end like that. Now, so that you can, if you now see, this is going to be U. Okay, this is UUC, UUC, UUA, then UUG. You need to learn how genetic code is being organized. Now, if you are to write on genetic code and you cannot draw this table, I don't know what you have done to that to justify that you really know it. So that means you should be able to draw. Go and look for formula how to crack this genetic code in case you come across questions on genetic code, so that you are going to do justice to it. It is very, very important. Now, after that, you now need to understand the process called translation. That is protein synthesis. What is a translation? You need to understand it very, very well. As how can you use these 64 genetic codons? How can you use it to be able to explain the process of translation? You should explain it very, very well. As is a protein synthesis. Please, I'm begging you, you should be able to know. As a student of molecular biology, you cannot run away from knowing, including single letter code, triple letter code, triplet codon, and how can you explain the process of translation. Translation also has its own modification, just like we have it in this. So it is very, very important that you should know that. Okay, and um, at that level, I want to actually give you like 1 minute, 1 minute break, so that I am going to write. Uh, so that what's going to happen is that you should be able to see what we have on the board, and you should be able to write it, jot it down. So please, it's very, very important. Okay, yeah. Okay, now let's go to another very important aspect of this course, and that is gene transfer. Gene transfer. I'm going to, because of the time, I'm just going to explain the three concepts. The three concepts with just maybe a few diagrams, so that you will be able to know exactly what you mean. So the next thing is transformation, conjugation, and transduction. I explain the, there are two types of gene transfer. We have horizontal gene transfer and we have vertical gene transfer. Anytime you come across vertical, vertical, that is from north to south, so that is from parents to their offspring, so that is a vertical. And anytime you now talk of a horizontal, that is like this, that is no, that is east to west or west to east, as the case may be. And when we say west to east, that is talking about um, the same species transfer, the same species or related species of uh, bacteria or microorganism. Now let's imagine an hypothetical example, okay, of a bacterial cell. Hypothetical example of a bacterial cell. Okay, so from this point, I want to explain with this name diagram, I want to explain the principle of what? Principle of conjugation, transformation, and as well as a transduction. Okay, now, okay. Now look at, I want you to look at this diagram. So the first thing is I want to look at transformation. Transformation. So that is this is going to be mechanism one or case one. Case two is going to be transduction. Okay, then case three is going to be conjugation. This is transduction. So the first one is a transformation. Transformation from the word transform, that is transform. We are talking of dead, naked DNA from the environment. Transform, dead, but the bacteria, this bacteria A is dead already, but the DNA is available in naked form. So if it is available in naked form, the DNA can now get itself transformed from being dead into the host cell. That is transformation. Transformation, dead. You have to take note. Dead and naked DNA is very, very available in the environment. For example, now, where we normally pour our laboratory waste, where we normally pour it. So immediately after that, you see the whenever the nutrients is no longer available for the microorganism, the microorganism is going to, the growth curve is going to enter death phase. And when it gets to death phase, the microorganism will die off. When they die, although they are already dead, but they will still live. Some of them will still live. They are naked. Their DNA naked, and it's going to be available until that naked DNA has gotten a very competent bacteria that the naked DNA can now become integrated into. Okay, can now become integrated into. So let me try and differentiate. Okay, you can see, let's say this is the host normal DNA. Okay, so the source can come from the source that is from blue. You can see that this DNA is blue. So the source that is blue is coming from transformed DNA from naked. So it's going to become integrated like this. Then the second one, which is transduction, anywhere you come across transduct, that should tell you that it is going to be involvement of a phage, involvement of a virus. Don't forget that the virus, the only infective part of a virus is going to be the genetic material. These coats, this is just the coat that is going to fall off whenever this has a. So this one is going to be red. So that's why you have it like this. It's coming from transduction, process of transduction. This time around, transduct, phage is involved. Now, the next one that has to do with, they discovered that some bacteria, they now have what is called factor. They call them factors. Factor F, which is going to be factor plus and factor negative. The one that is a factor plus is the one that processes the DNA, and is going to now pass it into the factor F. So remember, this one is in green. So this green will now become integrated into this. So that we now have one. We have two. One is coming from transformation from dead naked DNA. The second one, which is this two process called transduction, is coming from phage. Then followed by the third one, which is coming from conjugation. That's coming from conjugation. So that is the whole process or the whole diagram that can really help you to be able to explain. You should be able to explain and give you the overview of what we mean by when you come across the word transformation, when you come across the word transduction, when you come across the word what? Conjugation. The word conjugation. bliss. Conjugal bliss, sorry. Conjugal, that is coming together of male and female. That is conjugal bliss. So, and that is a bacteria also do what is called what? Marriage, whereby they also marry each other and donate some factors for them. So this is the host DNA. This is the host DNA, so that by the time the host has gotten DNA from the tree, so this DNA can now undergo the process of replication, and the process of replication continues, and you now discover that the new organism that will be formed from here is going to be a product. It's going to be a product. It's going to be a variant that is going to have different behavior entirely, and that is exactly what is called microbial genetics. Microbial. In fact, this is the beginning. This is the beginning and the basic understanding of where we have a microbial genetics, because the DNA here, as you have it here, from the uh, three different processes, is not going to be the same as the one that is just from one source. So that is why we have microbial genetics, and that is exactly what we want you to prove by understanding the concept, the basic concept of genetic microbial genetics. I want you to take note of that. Now, generally speaking, just like I said, whenever you are trying to explain, I don't need not too much diagram. What you need is just very simple and basic. Very simple and basic thing that you can use to explain your this thing. Even if you are going to have a diagram like this, there should be a kind of notes, very very short notes underneath to tell us uh, what you have done, to explain to us what you have done. So, and I want to believe that by you going through the materials that you already have, as well as much as what you have already on the YouTube, and if you can also combine the knowledge, general knowledge about it. Microbial genetics is not that difficult. The only thing is that you need to have some. You have to be uh, you have to be grinded very very well in the basis. So, and the basis is just what we have done so far. So I want you to understand that. Although there are like one or two other things that we did not and we could not actually cover, because I want this video to be as precise as possible. That's why I don't want us to stay on it very very long. I just coming up to give you exactly what you will need in order for you to be able to pass any exam on microbial genetics. And don't forget that the tutorial we had, the contact hour that we had. So I want you to convert all this and make very useful application of it, so that at the end of the day you will just smile in microbial genetics. And so I want to wish you best of luck. Don't forget to like, share, and tell your friends uh, to also like and share. And I want in the comment section, you can easily give me, maybe send me a note of whatever might be your response. So we want positive response as well as even negative feedback. That's going to help us to be able to improve in our subsequent video. Once again, I want to thank you very much for being here today. Thank you and God bless you.